Imagine you have a wound, a damage, and the body has this excellent capacity to heal itself. But when the gap is too large, body just doesn't manage. So what we do with the implant, we provide temporary bridge so the cells can again migrate over the support and doing what they know to do is like growing and healing tissue. So it's very important that TissueSpan is really just a structural support that enables the cells to cross a gap that is too big for them to cross by themselves.
What happens when a scientific insight about how the body heals becomes a device you can actually implant in a patient? Today we're taking a slightly different turn from our usual bioprocessing focus and stepping into medtech. My guest is Eva-Maria Balet, co-founder and CEO of Regenosca, who left the research benches of EPFL to tackle soft tissue repair. We'll trace her journey from pure science to building a company. Let's dive in. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Evie, welcome to the Smart Biotech Scientist. It's good to have you on today.
Thanks you for inviting me. I'm looking forward to our conversation.
Let's start with this perhaps a bit controversial question. Evie, share something that you believe about translating life science research into real products that most people disagree with.
I think one belief I hold, maybe a little bit controversially, is that starting with an exciting scientific discovery is not often the best model to create real-world impact in life science. So there are typically two approaches. Either you discover something in the lab and then you look for an application, or you start with a clear clinical need and design your research around solving it. And I strongly prefer the second approach. For me, being patient-centric is key. So when you begin with a real unmet need, it gives your work direction and purpose, and it keeps you focused on meaningful outcomes rather than just elegant science. And it also really helps when things don't go as planned, which is often the case. So because you stay motivated by the potential impact on patients. And at Regenosca, that's exactly how we work. So from the very beginning, we built on interdisciplinary teams that included clinicians. So patient needs were embedded in the research from day one. And in my experience, that mindset makes all the difference in translating science into something that truly matters.
Tell us your story, Evie. How did you first start in science? What drew you into that and then led you to EPFL and eventually now to Regenosca? What were some interesting pit stops along the way?
I've always been fascinated by life science, which is why I choose biotechnology. And what really intrigued me was the idea that you can use cells or microorganisms as tiny factories and essentially program them to produce something valuable. And that combination of biology and engineering really captured my curiosity early on. And then during my PhD at EPFL, I applied this mindset to tissue repair, working on scaffolds, so temporary structures that support the body in regenerating tissue. And what fascinates me most was that sometimes by providing just the right support, you can significantly enhance the body's own healing capacity.
That's funny when you're saying that. I think we have a similar start of our career. I studied as well at EPFL, and I was sitting in this biotechnology class, and then the professor told us that we could produce monoclonal antibodies in CHO cells. And that also got my attraction. It's like, wow, this is amazing.
For me, it's the same. It is just fascinating.
What was the thing that eventually pushed you, shall I say, over the line from academic research into starting your own company? That's quite a different ballgame, isn't it?
So when I was at EPFL doing my PhD and my postdoc, I was always drawn to applied science, where there's a clear path toward clinical use. And what ultimately pulled me from academia into entrepreneurship was the desire to see how our work actually reaches patients. So the strong feedback from clinicians, along with the motivation of my co-founders, really enforced that decision. Today, I think I still benefit strongly from my scientific background, but I have learned to translate it into a broader context. So science taught me how to learn, how to deal with uncertainty, and how to iterate toward solutions. And those skills are actually incredibly valuable when you build a company.
Let's start out simple to understand what is the problem you're solving with your company. You're in the tissue repair space. What is the exact problem you're solving there?
So soft tissue repair is needed in many situations. So after trauma, after the removal of diseased tissue such as cancer, or simply when tissue weaken over the time and lose their ability to provide mechanical support. So today's surgeons, they have several options, but none of them is truly ideal. So on one side, there are synthetic meshes, like meshes used, for example, in incontinence treatment. These meshes provide excellent mechanical support, they are highly standardized and readily available, but the downside is that they remain permanently in the body, which can trigger inflammatory reactions or complications such as erosion. And on the other side, there are biological materials derived from human or animal tissue, and these are more biocompatible, but they come often in challenges in terms of standardization, scalability, and consistent performance because they depend on donor tissue. And if no suitable product is available, surgeons often have to harvest tissue from the patients themselves, for example, from the inner mouth. That means creating a second surgical site with additional pain, risk of complications, and also very limited supply of tissue. So despite many available tools, surgeons are still working with compromises, and that's exactly where innovation is needed.
And what kind of innovation did you do? What kind of tissue are you now developing?
So our implant we are developing is called TissueSpan. And at the heart of TissueSpan, there's a very simple idea. Instead of replacing tissue, we support the body to heal itself. So TissueSpan is a fully engineered collagen implant produced through a proprietary process that allows us to create a mechanically stable structure by preserving the natural bioactivity of collagen. And that combination is really key. So for the surgeon, it's very straightforward, an off-the-shelf, ready-to-use implant for soft tissue repair. And once implanted inside the body, the biology takes over. So cells from the surrounding tissue, they migrate into the scaffold, new tissue forms, and over time the implant is completely absorbed and replaced by the patient's own tissue. So in the end, there's no permanent foreign material left inside the body. And what makes TissueSpan fundamentally different is that it combines the strengths of current solutions without the limitations. So it's standardized and scalable like a synthetic mesh, but it integrates naturally like a biological material without relying on donor tissue or requiring a second surgical site.
How long does this process take place? So you put your collagen-based tissue inside the body, then cells come eventually populate that, and then eventually it's degraded. So are we talking about a few days? Is that several months, or how does that work?
So we are speaking about months. So once TissueSpan is implanted, cell migration, cell infiltration starts straight away. And then it takes 3 to 4 months until the implant is completely absorbed. So it's really this timing about cell ingrowth, regrowing new tissue, and the absorption.
What are the underlying mechanisms that once you have put that into the body, that cells migrate into that? Is that biological? Is that something else? Or how does that work?
Imagine you have a wound, a damage, and the body has this excellent capacity to heal itself. But when the gap is too large, body just doesn't manage. So what we do with the implant, we provide temporary bridge so the cells can again migrate over the support and doing what they know to do. It's like growing and healing tissue. So it's very important that TissueSpan is really just a structural support that enables the cells to cross a gap that is too big for them to cross by themselves.
Oh, that's brilliant. That's highly fascinating. So the biological process itself does work. It's just too big of a step if I translate that into very simple terms.
Yeah, that's how I would also say it.
Excellent. And how do you produce these collagen-based implants?
So you want to know a little bit more about manufacturing?
Yes.
Okay. So manufacturing is actually like a very sensitive topic for us. So I will stay on a high level. So TissueSpan is based out of a molding technology. So we start with collagen as a building process. It goes into our molding process, and then at the end we end up with a mechanically stable implant that is ready to use, sterile, and provided ready to use to the clinicians.
Translating now this scientific discovery you have made into a real physical device, well, as we all know, is quite a challenge. What were the biggest hurdles, whether it's on a technical, scientific, or even on the perhaps more an entrepreneurial side? What are some of these hurdles you have faced?
So bringing a scientific concept from the lab to something you can safely use in patients is indeed a major step, and it requires a complete shift in mindset. In research, the focus is on providing a concept, but for clinical use, everything needs to be reproducible, controlled, and aligned with strict regulatory requirements. So one of our biggest challenges was translating our lab process into a robust and scalable manufacturing process. So that meant moving into a GMP environment and implementing full control over every step, like from raw materials to process checks and final product testing. So ensuring full traceability and consistent quality. It also required building a highly interdisciplinary effort. So we had to combine expertise from scientists, engineers, and quality and regulatory specialists to meet clinical and regulatory expectations. And another key challenge was preserving the unique properties of our material while adapting the process to industrial standards. Because what works in the lab must work reliable every time you produce it for a patient. And in the end, the innovation in medtech is not just about the science. It's about making that science reliable, scalable, and safe for every single patient.
I'm curious, your product, with respect to the regulatory requirements, it's considered as a medtech product. So I imagine the regulatory requirements are much more strict than, for instance, for a biological drug, or is that similar?
It's like two different worlds. You have medical devices and you have drugs, and each world has its own regulation and standards to follow. But that's also one thing I actually appreciate about the regulatory space, because it provides like a structured framework with clear definitions and guidelines that help you classify your product. At a high level, like a medical device is defined by the primary mode of action being physical or mechanical, whereas drugs and biologics act mainly through pharmacological, immunological, or metabolic mechanism. And in our case, the primary function of TissueSpan is structural, which clearly positions us in the medical device space. And I think it's a useful exercise to go through this classification yourself as a founder, because it really forces you to understand your product in depth. But it's equally important to validate your interpretation early with experts, whether that's regulatory specialists, notified bodies, or authorities like Swissmedic. And we are very fortunate because one of our co-founders brings strong regulatory expertise and leads this area. He has very good knowledge and is also supported by a broader network when needed. And that has been extremely valuable in navigating this pathway efficiently.
This is quite a challenging undertaking, especially in a startup environment where you not always have all the expertise at your hand. So I would be curious, Effie, what would you tell a founder who is listening, who's also trying to figure out how to navigate the regulatory landscape? What are some pieces of advice you could give them? What worked well for you in this process?
I think what worked extremely well for us in the process was to have this in-house expertise of regulatory, because one of my co-founders is a regulatory consultant. He is used to navigate guidelines, standards, and he has like also this overview, what standards and which guidelines are available. So he can really help us to find the best and most efficient pathway. But I think if you don't have it in-house, there are a lot of regulatory consultants out there which create expertise. So then I think it's very important to find your regulatory consultant of choice that you take along with you on the journey, and you build a strong relationship so that he can support you as good as possible.
Yeah, that's definitely important. A leadership principle I love to adhere to is who, not how, which means it's much better to have the right people. So in other words, the who's on your team, or as you say, if not possible, as a consultant or otherwise, then figuring out all the complicated things yourself, especially on the regulatory side. It's quite a difficult world if you don't have the expertise yourself, and mistakes can be very costly.
No, I completely agree. And it's like no founding team can cover everything. So that's why, as you say, it's important to build a broader ecosystem around you, whether that's advisors, early team members, or board members who can actually complement your skills and bring additional expertise. For us, the team composition was actually both intentional and organic. So before founding Regenosca, I have already been working closely together with my co-founders in the lab. So there was a strong foundation of trust and mutual understanding and confidence in each other. And what really united us was this shared belief in the technology and the deep collective understanding of it. But we had all a different perspective. Like we had this clinical perspective, regulatory perspective, scientific and entrepreneurial science perspective. And I think this interdisciplinary is really one of our core strengths.
The leap from a lab bench to a device inside a patient is rarely a straight line. It's built on inflection points, hard technical problems, and a clear view of what's failing patients today. Eva-Maria's perspective on that path is one you'll want to carry into part 2. If this episode added value, please leave a review on Apple Podcasts or your favorite podcast platform. Thank you for tuning in, and I'll see you in part 2. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.